Alright, so once your second round of classification is over, and you have these lovely black and white binary masks, admittedly these are kind of concerning looking because I only did a couple rounds of classification there when I was training with classifiers. But you have these binary masks, and you can see even with, like, I think I maybe spent 15 minutes on the training, they're still pretty good, especially in the middle here. And I guarantee, especially if you go and look at the actual results of the book chapter, you get much better classification than this. But just for the tutorial and for the sake of understanding what we're doing, this is sufficient. We're going to go ahead and get to the tracking section. So it's going to ask the minimal cell area for tracking. So we're not going to track anything above or below, anything below 100. If you want to know like what area around you should be putting in here, you can actually measure the area using ImageJ, and that's how we were doing things earlier. So you just click and load a file and say if there were like something out here that you knew wasn't the cell or something, or you can just go and measure one of your cells and see what the general typical area is. You can go to Analyze, Set Measurements, make sure area is checked, and just use that bean shell tool we're familiar with, and maybe pick a kind of smallish cell like this. Kind of see what the area is. You can hit Control M or go to Analyze Measure. See, this area is 95, so it actually wouldn't track this cell. So maybe if instead of doing 100, we might do something smaller like 80. And that's going to ask the maximum cell area, which I probably should have gone and found a rather large cell to measure. So like here, these cells are rather large. So it's going to measure these as a cell that hasn't divided yet, so I might just kind of do that. Hit Control M and see how this is a 433. So you can see 2500 is quite large. We're going to use it. And the minimum number of frames to track cells through. The maximum number of frames you can have is 2 less the total number of frames available. Simply because that's what's available. So we're going to say 15 because we only have 20 frames. So the maximum number is 18. 2 less 20 is 18. And we're going to say 15. Do you wish to analyze a region of interest? This now is asking for the sake of whole image analysis and video render. And you can actually, instead of analyzing the whole image, you can additionally just analyze a region of interest done using the same process we did to make an ROI file earlier. So I'm going to say yes. And then do you want to crop an image based on the ROI? These have to be the same ROI file. By the way, I'm going to say yes. So in the video, it'll be cropped to whatever ROI I specify. And then you need to enter the path to the ROI file, which means we need to make an ROI file. So again, I'm just going to go ahead and open up an image. I'm going to make sure my measurements are only a bounding rectangle. I'm going to remove the scale. And then we're just going to go ahead and select a region of interest like so. And hit Control M. Or measure. Apparently the results table is hidden behind something. There it is. We're going to close the results table so that we only have the one measurement we are actually interested in. And we're going to save that as a CSV file. Again, it doesn't remember where I want to be saving stuff, so we want to save that in our working directory. And I just like to call mine ROI. You can name it whatever you like. Go ahead and close that out and tell it we're looking for ROI.csv. And this is, do you want to number the cells in the image based on lineage tracking? So your final video will actually have little numbers on all of your cells. It can be kind of like confusing looking, but I think it's useful for having like a visual output. So I'm going to say yes. Also, do you want to outline the cells based on the mask? So if you want to know how good your segmentation is by overlaying that onto the original images, you can say yes here. And I'll go ahead and do it. I said no simply because having both the numbering and the outlines can get kind of confusing. And this process will take a while, the tracking. It'll just keep reading out things and telling you what step of the process is at. And we'll come back when it's done.